From this presentation, we are going to learn about the various plant sources available for the preparation of drugs. A huge number of plants have medicinal qualities used for centuries as drugs or drug sources. So they have been exploited for the preparation of various drugs used in therapy. The earliest plant source for drugs was the leaf. In ancient times, our ancestors used the leaf of a plant to prepare the drug for medicine. Other parts of the plants, example barks, fruits, roots, stem, wood, seeds, blossoms, bulb, etc. They are also used for the preparation of drugs. Then let us see what is meant by crude drug. The product used without further processing. For example, ground leaves of a plant or a bark, boiled concoctions or powdered sap is called as a crude drug. So that means that a part of the plant used in a crude form without any processing step is called as a crude drug. So this is an image showing the preparation of a drug from the plant source. Now we are going to see various animal sources used for the preparation of drugs. Many important drugs are animal derived. Medicinal substances are derived from the animal's body secretions, fluid or glands. Therapeutically effective materials or therapeutically efficient chemical substances are present in the secretions or fluid or secretions of the glands of the animals. For example, insulin, heparin, adrenaline, thyroxine, cod liver oil, musk, bee wax, enzymes and antitoxin sera. They have been prepared from the various animals. And this is a table showing the animals, various animals from which various medicines have been prepared used for treating various disorders. For example, the pancreas of the cows, we can isolate insulin, which will be the anti-diabetic hormone, and the sperms of the fish, protamine sulfate is being prepared, that will be the antidote of heparin, and intestine of pig, from the intestine of pig we will be able to prepare heparin, and that will be a useful anticoagulant, and from the lungs of the ox we can get heparin, that will be again an anticoagulant used to prevent the clotting process in the blood circulation. Similarly, we can prepare the various drugs from microbial sources also. For example, penicillin is being produced by Penicillium chrysogenum, and streptomycin is prepared from Streptomyces griseus. Chloramphenicol is prepared from Streptomyces venezuelae. Neomycin from Streptomyces fradii, bacitracin from Bacillus subtilis, xanthan from Xanthomonas competitris, dextran from lactic acid bacteria, leuconostoc mesenteroids, Streptococcus mutans, Lactobacillus brevis, and cardian is prepared from Agrobacterium biobar and Alcaligenes faecalis, and pullulan is prepared from fungus Basidium pullulans. So these are all the various drugs prepared from the microbial sources. And this is an image showing the microbial preparation of medicines used for treating many disorders. Marine sources. So we can also see the various marine sources available in nature to prepare various drugs. For example, we can get the drugs from coral, sponges, fish and marine microorganisms, which can produce biologically efficient or potent chemicals, which has an anti-inflammatory, antiviral and anti-cancer activity. So to treat the infections, to treat cancer or inflammatory condition, we can make use of the drugs prepared from various marine sources. For example, Curacin A is derived from marine cyanobacterium Limgia majuscule, and Eleutherobuin is obtained from coral Eleutheroboia species, And discodermolyte is obtained from marine sponge Discodermia dissoluta. And this is an image showing how the drugs are being derived from the various marine sources. And we can also get the drugs from the minerals which are available, which are distributed in nature surplusly. And our body requires trace elements of minerals to maintain the hemostasis. So that means that we should maintain a balance of the mineral in our body in order to meet out the requirements, mineral requirements of our body. In case if a particular individual is deficient of a mineral, he has to be supplemented with the specific mineral. So to treat such patients, we can make use of mineral based drugs to raise the level of the mineral in order to treat the deficient cases. And we have got enormous mineral sources to treat various disorders. So here is the list of the minerals used in treating many disorders. For example, ferrous sulfate is used in treating iron deficiency anemia, and magnesium sulfate is used as a purgative, and magnesium trisilicate, aluminum hydroxide and sodium bicarbonate are used as antacids used to treat the peptic ulcer. Zinc oxide ointment is a skin protectant in wounds and eczema. Gold salts are acting as anti-inflammatory and in rheumatoid arthritis, and selenium is used as an anti-dandruff agent. And radioactive isotopes of iodine, phosphorus, gold are used for diagnosing and treating various malignancies. And there are synthetic and chemical derivatives, nothing but the drugs used to treat various diseases. A synthetic drug is produced by a chemical synthesis, by chemical means. By having a proper, by following a proper laboratory procedure, we will be able to make the chemical derivatives, nothing but the drugs in a synthetic form. These drugs have higher yields with the quality, purity and low cost. So it is better to produce many drugs in order to meet the demand. And various synthetic or chemical derivatives are evolved with human skills in the laboratory and advanced knowledge and understanding of phytochemical investigation. Majority of drugs are used in clinical practice, prepared synthetically in pharmaceutical and chemical industries and laboratories. One of the earliest synthetic drug was sulfonamide. Other examples are acetyl salicylic acid, nothing but aspirin, oral antidiabetics, antihistamines, thiazide diuretics, chloroquine, chlorpromazine, general and local anesthetics, paracetamol, phenytoin, etc. And another more important technique, which is called as recombinant DNA technology, is also being exploited to prepare various drugs in order to meet out the demands. Recombinant DNA technology enables the production of wide range of therapeutic agents in sufficient quantities, which can meet out the demands for the medicines in society, in human society. The commercial exploitation of recombinant DNA technology began in late 1970s by biotechnological companies to produce proteins. There are around 400 different proteins are being produced by rDNA technology, recombinant DNA technology, and only 30 were approved by the Medical Council for human use. And there are three types of the products obtained by recombinant DNA technology. The pharmaceutical products of rDNA technology are broadly divided into following three types. Number one is human protein replacements. Number two, therapeutic agents for human diseases, vaccines. So we will be able to produce human protein replacement to treat the protein deficiency conditions, to treat the protein disorders. And there are therapeutic agents which can also be produced by using recombinant DNA technology to treat various human diseases. And there are vaccines that can also be produced through recombinant DNA technology to prevent many infectious diseases. Thank you for your patient hearing.